SSCP stands for single-strand conformation polymorphism and is used to demonstrate the presence of mutations. By SSCP you have two double-stranded DNA segments, one wild type and one possible mutant. Mostly these DNA segments are products of a PCR with a length of a couple of hundred base pairs. Next up, the DNA segments are denatured at 95 degrees Celsius, and the DNA strands come apart into single strands. After this, the temperature is lowered rapidly, whereby the single-stranded DNA segments fold in itself. The rate of temperature reduction is playing an important role in the prevention of the annealing of the segments onto each other. Because the existence of a mutation in two of the segments, these segments fold in a different way. The next step is to perform a gel electrophoresis. This is done with a molecular marker and both of the samples. If the samples were unfolded, they would look the same size, but in the folded state they will migrate differently, trough the gel and end up at a different position. Thereby, if samples differ from the position of the wild type, they have a different 3D shape, which is caused by a mutation. If SSCP is used in combination with capillary electrophoresis, addition probes need to be added to the double-strand DNA strands. For example, FAM can be attached to the 5' side of the leading strand. Hex can be added to the 5' of the reverse strand. These probes will make the detection by the capillary electrophoresis possible. Everything else remains the same, except for the detection method. Here you will see signal peaks for when the single strands pass by the detector. Because the 3D frame of the samples are different, the location of the peaks will be different as well. Here we present the differences between GE, gel electrophoresis, and CE, capillary electrophoresis. First of all, the GE is cheaper to use. The samples are reusable, but is less accurate. The CE is very accurate, but uses expensive probes and the samples are lost after processing. In conclusion, SSCP detects the existence of polymorphisms, but not their location. We think this technique is especially useful for pre-selection of large quantities of samples with possible mutations, to narrow down on mutated samples and make a project overall cheaper. An alternative technique that will give the same result is melt curve analysis. Finally, to really know what kind of mutation is present and on which location, sequencing is necessary. But if you don't need that information, just conformation of a polymorphism, this technique is rather easy and cheap in use. Thank you for your attention. Goodbye. Have a nice day.